The overall goal of this procedure is to virally manipulate oligodendrocyte precursor cells, or OPCs, to overexpress proteins of interest for the in vitro study of CNS myelination. This is accomplished by first cloning a lentiviral vector. The next steps are to produce the lentivirus and determine the optimal viral concentration for the experiment. Following this, dissected and cultured OPCs are transduced with the lentivirus. The final step is to seed the transduced OPCs onto cultured neurons, dissected from the DRG. Ultimately, the myelinating co-cultures are assessed for myelin protein expression by Western blot analysis and visualized for the formation of myelinated axonal segments by immunostaining.
The main advantage of this technique over existing methods, such as pharmacological tools, is that the lentiviral approach allows us to selectively overexpress either wild-type or mutant proteins in our desired cell type, such as, in this case, oligodendrocytes.
In preparation, clone the required 2K7 lentivectors as described in the text protocol. In the morning of the day of transfection, prepare 32 million HEK293 T cells in a T175 flask containing 25 milliliters of media. Critically, these cells must be stuck down before transfection, so plate them sooner if needed. In a 50 milliliter Falcon tube, prepare a master mix.